Transformation. Transformation is a mechanism of horizontal gene transfer in which bacterial cell takes up free or naked or soluble DNA fragments from its surrounding environment but and incorporates them into its own chromosome in a heritable form. It was the very first example of genetic exchange in bacteria to be discovered. The mechanism of transformation for successful transformation, the naked DNA must first be absorbed by the recipient cell, enter the cytoplasm, and then undergo recombination with the host genome. The size of the DNA fragment affects its transforming ability. Typically, DNA with a molecular weight of less than 0.3 million Dalton fails to transform the recipient cell. Natural versus artificial transformation. Natural competence. Only certain genera of bacteria are naturally competent, meaning they naturally possess the ability to take up DNA from their environment. Examples of naturally transformable bacteria include Bacillus subtilis, Haemophilus influenza, Neisseria gonorrhoeae, Streptococcus pneumoniae, Acinetobacter, Pseudomonas. They're all the natural competence examples. Artificial transformation, we call transfection. Some bacteria, such as Escherichia coli, resist transformation under normal conditions. However, they can be artificially induced to take up DNA through a process sometimes called transfection. This involves subjecting the bacteria to special treatment, such as exposure to calcium chloride, which alters the cell envelope and makes the bacterium permeable to foreign DNA. Historical discovery of transformation. Griffith's experiment, the concept of transformation was directly proven by Frederick Griffith, 1928, through his classic experiments with Streptococcus pneumoniae or Pneumococcus pneumoniae and mice. Griffith noted that injecting mice with live and non-encapsulated rough a virulent bacteria or with heat killed encapsulated smooth or virulent bacteria did not kill the mice. However, when he mixed the live non encapsulated bacteria with the heat killed encapsulated bacteria and injected the mixture, the mice died. Griffith was able to isolate live encapsulated bacteria from the blood of the dead mice. He concluded that a transforming principle from the heat killed bacteria had entered that live virulent cells, genetically altering them to synthesize a capsule and become virulent. Griffith experiment of transformation. Griffith noted that injecting mice with live non encapsulated bacteria or with heat killed encapsulated bacteria, mice did not kill. Means when we inject non encapsulated live bacteria, mice did not kill. Heat killed encapsulated bacteria, mice did not kill. But when he mixed the live non encapsulated bacteria and heat killed encapsulated bacteria and injected the mixture, the mice died. That means Griffith was able to isolate live encapsulated bacteria from the blood. That means there is something transformed from heat killed encapsulated bacteria to non encapsulated bacteria. So he concluded that a transforming principle from the heat killed bacteria had entered the live virulent cells, genetically altering them to synthesize a capsule and become virulent. In 1944, scientists Oswald T. Avery, Colin MacLeod, and Macklin McCarty built upon Griffith's work and definitely identified that this transforming principle was actually deoxyribonucleic acid, DNA, that which Griffith not able to say that, but these people transforming principle that they proved that deoxyribonucleic acid. Oswald Avery and Colin MacLeod and Macklin McCarty. Significance of antimicrobial resistance. While the transfer of antibiotic resistant genes via transformation can be easily demonstrated in laboratory experiments, its actual significance and frequency in the natural spread of drug resistance among bacterial populations remain unknown.